Okay, we are back in the TYBSc Zoology Mammalian Histology course. Previous lectures of a liver was interrupted, so we start from that point only. So here it is a cross section of a hepatic lobule. Here they have shown a cross section of hepatic lobule. Here is a central vein, and this is a liver parenchyma. In that liver parenchyma, there is a gaps between the hepatocytes forming the hepatic sinusoids. Okay, and these are the sites or area of a hepatocytes called as hepatic plates. Then these gaps are filled with the blood coming from the hepatic artery and hepatic portal vein. In uh, another side of these sinusoids around the hepatocytes, each hepatocyte is provided with the thinnest possible capillary-like structures of a bile canaliculi, which collect bile from the each hepatocytes pour into these small micro canaliculi, and they collect together and open it that pour that bile into the bile ductule. So this is a portal triad, a union of a location of three vessels at the corner of hepatic lobules. This is a hepatic lobule, this is central vein, and these are all sinusoids. Okay, so these are all hepatic sinusoids. I think now you can imagine how that hepatic sinusoids are there. Okay, in this actual color photograph of a hepatic of a liver, you can now easily identify that this is a central canal. It's a central canal of a hepatic lobule. So if we consider this all radiating cracks at the central portion here, so this could be a single hepatocyte or hepatic lobule. This is a single hepatic lobules directed centrally, and this is a central vein. So at the corner here, these are the portal triad are there. These are the portal triad. Why? Because here you will see opening of three different tubules or vessels, that is artery, vein, portal vein for that hepatic artery, and the bile duct. So these are the portal triads, and this is a central vein, and all these are the hepatic lobules. Now all these cracks, what we saying as a crack here, white color crack, is a hepatic sinusoids, a space between the line of two hepatocyte cells. Okay, here in a hematoxylin eosin stained color photograph of a liver slide under microscope. This is a central vein. All these whitish line indicates the sinusoids, and all these pinkish color structures with a dark prominent nucleus, dark color prominent nucleus. So all these are the hepatocytes, and these white color structures are the hepatic sinusoids. So here this is a magnified view of a portal triad. So here is a artery because there is accumulation of RBCs and lot of vascular, a lot of muscular layer you see here. So that is the artery, and here this is a bile ductules because there is a cuboidal epithelial layer is there, and less amount of circular muscle. So this is a bile canaliculi or duct, and somewhere here there must be some portal vein is there. Okay, now diagrammatically, if you want to arrange that hepatic lobule, if you want to understand the arrangement of hepatic lobule, it is like this. So if this is a one hepatic lobule, hexagonal hepatic lobules, all these are the sinusoids running towards the central veins. All these corners are with the that triad. Now how these triads are spread in a hepatic lobule, so they produce these branches, and these branches then pour their blood, and the blood goes to the central vein. But these canaliculi, all these canaliculi collected towards and form a single bile duct, and they pour the bile into the bile duct. Okay. So this much area, from this much triangular area, these biles is collected to the triad, and here from these periphery portions of hepatic lobules, this um, blood pour and goes to the central portion. So these triangular portions represent the exocrine part of this liver, and this is the endocrine portion because a lot of secretions take place here. Metabolic activity goes on in there. Now again, a magnified views of this triad, how that triad are communicating with the hepatocytes, that is shown in this diagrammatic presentation. In this color photograph, if this is a portal vein, then this is a hepatic artery, and this is a bile duct tubes, and these are the lymphatic vessels. Okay, all these are the hepatocytes, then hepatic sinusoids are there. Now here in this diagram, you can see here how bile ducts are formed. Now if we consider this as a single hepatocyte, this dark grayish color structure, dark brownish color structure, if you consider these all are the hepatocytes. Now see, the cellular component of a hepatic lobule or liver is what? These are the prime functional unit hepatocytes. Okay, thereafter these are the blood vessels, capillaries, though we call them as a hepatic sinusoids, but basically they are discontinuous capillary. If you remember that capillary lecture, there we said about the discontinuous capillary. So these are the discontinuous capillary. Now here it is called as a hepatic sinusoid. So all these cells are the endothelial cells. They have very large gaps in between, so that blood can reach and touch to these hepatocytes, and all that metabolic detoxification, everything happens in the hepatocytes. Now simultaneously, another side of this hepatocyte, one side is towards the blood and another side is open towards this gap. These gaps are called as a bile canaliculus, or the larger gaps are called as a cholangioles. These are related with the bile secretion. So in these gaps or sinuses, this bile secretion take place, and it is these are the cuboidal epithelial cells lining, and so they form the tube system here. Because of this epithelial tissue, they form the tube system here, and these bile canaliculi come together and forms a larger bile ductules, and that before the bile ductules, the tube is called as a canal of herrings. So overall, the cellular system in a liver is endothelial cells, hepatocytes, then bile tubules lining cuboidal epithelial cells, and along with that you will find one more type of phagocytic cells spread in this sinusoid. They are the Kupffer cells. Okay, so these are the nucleus. Now here again they have shown the diagram representation of a sinusoid concept. See all these hepatocytes and the green dot. This green dot is a junction between the two hepatocytes. They are not directly come in contact with the blood or sinusoids, but they are in close contact with between the two hepatocytes. There is a gap, and through that gap that form the bile canaliculi, and all that bile is poured into this bile canaliculi, and again then they forms the cholangium, and they form that duct of herrings, and then lead to the bile duct. Yeah. And in between the sinusoids, which is lined with the endothelial cells, this blood flow, and that blood then come in contact with the hepatocytes, and there take place the exchange of nutrients and metabolism and rest of the thing. So just take a look to this labeling. This space between the endothelial cell and the hepatocyte, that space is called a space of Dice. And these are the liver sinusoids. These are the sinusoidal endothelial. Then is a central vein, then hepatocyte or parenchymal cells, then bile canaliculi. Now in this photograph of HE stain liver section, here you can confirm that what is shown in this diagram. So this nucleus indicate the nucleus of hepatocyte. These are the hepatocytes. Okay. Then these are the hollow portion. They are the hepatic sinusoids, and this is a Kupffer cells. And this junction, if you can see here, it is a canaliculi, bile canaliculi. Okay, so this you try to see only the space here, sinusoids, hepatocytes nucleus, and if you see here, this is an additional cell than the hepatocyte in a sinusoid. These are the Kupffer cells. They are phagocytic cells. The role is in a like WBCs, macrophages. Now this is a liver vasculature. Here they have removed every parenchymal cells except the blood and blood vessels. It look like this. All these are the embedded blood vessels. This continuous capillary entered into the parenchymal portions of a hepatic lobule. So if you consider this as a hepatic lobule, then these are the triad or larger arteries and veins, and their branches in the form of a sinusoids enter into the hepatic lobules, and this is a central canal here. Now here liver parenchymal cells. Okay, this is a scanning electromicrograph of a liver parenchyma. So you can clearly see or understand the concept of sinusoids and the parenchymal hepatocytes and central vein and this artery and the portal veins and this bile duct. Here again the hepatic sinusoids is clearly. This is the gap through which the RBCs are flowing, that means blood is flowing, and all these surrounding are the hepatocytes. Another side of this hepatocyte, here there is a bile canaliculi. So here again the sinusoidal discontinuity, or you can say the discontinuous capillaries are that means the sinusoids. Okay, sinusoidal discontinuity to ooze out that blood and reach to the hepatocytes. Okay, now hepatic parenchyma cells. These are the Kupffer cells, macrophages. Now in this diagram, they have shown a single hepatocytes and surrounding structures around the hepatocytes. So hepatocytes, they have a few surfaces like this area towards the sinusoid is called as a sinusoidal surface, and this area towards the bile canaliculus, that is called as a canalicular surface, and intercellular surface, if that hepatocytes has a connections with the another hepatocyte, then that surface is called as a intracellular surface. Okay, but here they have not shown any attachment of another hepatocyte. So if it is attached to the another hepatocyte, then it is an intracellular surface. So here clearly visible is a canalicular surface and a sinusoidal surface. So this is a hepatocyte. Here all cell organelles you can see clearly. A lot of Golgi complex, then mitochondria, endoplasmic reticulum, everything, lysosomes, and then glycogen granules. When that glucose converted into glycogen, it is stored into the liver cell. So you will find all these glycogen granules stored in them. Okay, these microvilli structures on the surface of a sinusoid, mostly when it is towards the sinusoidal surface, and these are the endothelial cells of a capillary sinusoidal endothelial cells, and outside this there is the Kupffer cells, macrophage cells. And so through these gaps, through these microvilli, all that exchange and metabolic activity take place with the hepatocytes. Okay, these are the some of these are the ETO cells or fat storage cells. They develop between these microvilli and the endothelial sinusoidal structures. So in case of clinical conditions like fatty liver, Majority of these sinusoidal surfaces are blocked because of fat accumulations, and the liver functioning get reduced. So it is called that this liver is a fatty liver, and the functioning of liver is reduced. So as soon as this fat get removed from this area, the functioning of liver increases naturally because of more and more blood come in contact with the hepatocytes. There is no barrier for fat storage there. Forget it. It's a clinical thing. We are dealing with the classic histology. So here the basic thing to know is a hepatic sinusoids and hepatic sinusoidal surface, then sinusoidal endothelial lining, then Kupffer cells and the hepatocytes, and this bile canaliculi. So all whatever the bile produce, it is poured into bile canaliculi. This is a single hepatocyte here. So in large nucleus, all these cell organelles are clearly visible here. Mitochondria, smooth endoplasmic reticulum. This is sinusoidal space, and this is the bile canaliculi area. This is the bile canaliculi surface. Okay, now if we want to see whether really these glycogens are stored in a hepatocytes or not, then you use this path staining technique, and that way you can highlight the presence of glycogens accumulated into the hepatocytes. All these violet color spots are glycogen granules accumulated into the hepatocytes. Okay, so this way you can see under electron micrograph, glycogen look like this dark spot in a hepatocyte. This is a single hepatocyte cells with lot of deposition of these glycogens. Then endoplasmic reticulum is highly developed in case of hepatocytes. You will find lot of endoplasmic reticulum. Then these peroxisomes, then smooth endoplasmic reticulum are clearly visible in a liver hepatocytes section. Now bile canaliculi, again just to revise the concept of how bile canaliculi are communicating with the hepatocytes. So you see this border is entirely lined with this bile canaliculi, and all that bile flows through this canal. So every hepatocyte surface is surrounded with the bile canaliculi, and this flat surface in contact with the blood, and this side surface is in contact with the bile. So here by using a particular stain, you can see the presence of bile canaliculi communications with each and every hepatocytes. This bile canaliculi they are tried to show here. So this round pore-like structure is a communication pathway of this bile canaliculi. So in between the junction, wherever you see this round bead-like structure, that is a bile canaliculi communication. So here again you can see a bile canaliculi. Okay, here again you can see bile canaliculi. So all these are the bile canaliculi communications pathway here. All these are the hepatocytes, and these are the sinusoids. See in the electron micrograph that bile canaliculi structures around hepatocytes look like these lines. These lines surrounding the hepatocytes are bile canaliculi. Here again you can see the groove-like structures through which the bile flow, and they are very close in contact with the hepatocytes. Here again this is a tight junctions thing, bile canaliculi. This is a gap junctions. You will learn that in our next classes. Okay, this is freeze structure, freeze fracture system for electron micrograph. There again you can confirm the presence of bile canaliculi. Then Kupffer cells. Now whether really there is a macrophages present in a liver, so they have used a particular carbon in dye injected into the liver, and they notice the presence of all these black color spot. All these black color spots are specific Kupffer cells. This is a central vein. This is all that hepatic lobules. These are the triad. Here is a triad, and all these radiating white color fractures represent the sinusoids. In between, you will see here these black spots are the Kupffer cells. Then here Kupffer cells. These are the wandering macrophages like WBCs. So they move in a sinusoids, and if they found some toxic material or foreign thing, they engulf it and serves as a WBC or Kupffer cells, macrophages in a liver. All these are the Kupffer cells here.